The complement system is a complex cascade composed of over 50 proteins that play an important role in various biological processes, including its primary role, facilitating host immune response to invading bacteria, viruses, fungi, and parasites through opsonization, inflammation, and direct lysis, supporting cellular clearance and recycling via apoptosis and autophagy, contributing to neural development through synaptic pruning, with more functions being discovered regularly. Given its biological importance, complement system dysfunction is now associated with numerous localized and systemic diseases, such as atherosclerosis, cancer, neurological disease, kidney disease, immune system disorders, and trauma. Multiple complement-specific therapeutics are currently available, with many more in clinical trials and development. This video will detail the process of complement system activation, its role in various disease states, and laboratory tools available to further study its role in human biology. Let's explore these functions in further detail. Complement system initiation. The central protein of the complement system is C3. All initiation pathways converge at C3, which is then cleaved into fragments C3a and C3b via protein complexes called C3 convertases. The complement system can be initiated by three canonical pathways: the classical pathway, lectin pathway, and alternative pathway. Each recognizes different cellular features, providing comprehensive immune detection. The classical and lectin pathways utilize similar processes to initiate activation. The classical pathway. The classical pathway is typically activated by antigen-antibody complexes, although there are other activators of this pathway. The pathway begins with C1, a macromolecular complex composed of C1q and 2 units each of C1r and C1s. C1 binds to the Fc portion of multiple IgG antibodies, or a single IgM antibody, initiating a conformational change in C1q and subsequent conversion of C1r and C1s into their active serine protease forms. The activated C1 complex then cleaves C4 into C4a and C4b, and C2 into C2a and C2b. The lectin pathway. The lectin pathway is activated by specific carbohydrates common on the cellular surface of pathogens. Similar to the C1 complex, mannose-binding lectin, MBL, combines with MASP1 and MASP2 proteins, binds to cell surface carbohydrates, and activates them as serine proteases. These enzymes cleave C4 and C2 just like in the classical pathway. In both pathways, C4b covalently binds to the cell surface. C2b then binds to C4b to form the classical and lectin pathway C3 convertase. The alternative pathway. The alternative pathway is initiated by the spontaneous hydrolysis of C3 into C3 water, which binds protein factor B. Protein factor B is then cleaved by factor D into fragments BA and BB. Fragment BB stays bound to C3 water, creating the soluble C3 convertase of the alternative pathway. C3 convertase complexes. The C3 convertase complex created by the classical lectin and alternative pathways can all cleave C3 into C3a and C3b, and the complement cascade can continue. Complement system amplification. Once C3 is cleaved, fragments C3a and C3b have different purposes. C3a is a powerful anaphylatoxin that recruits additional immune system functions. C3b is a strong opsonin and can bind directly to cell surfaces. Membrane-bound C3b can then bind to factor B, which is then cleaved by factor D into BA and BB. This C3b-BB complex forms an additional C3 convertase, creating a complement system amplification loop of continuing C3 cleavage. The terminal pathway. C3b can also bind to existing C3 convertases to form C5 convertases, which cleave C5 into fragments C5a and C5b, initiating the terminal pathway. C5b binds to the cell surface and recruits complement system proteins C6, C7, C8, and multiple C9s to form the terminal complement complex TCC, or membrane attack complex MAC. MAC forms a pore in the cell wall, allowing for an influx of extracellular solution via osmosis, ultimately leading to lysis and cell death. Complement system immune system recruitment. After initiation and during amplification, the complement system releases powerful anaphylatoxins. The C3a and C5a generated by the C3 and C5 convertases act as chemoattractants, recruiting immune cells such as macrophages, mast cells, and basophils, while also inducing degranulation and release of histamines and other vasoactive amines. Together, complement system initiation, amplification, lysis, and immune recruitment provide a comprehensive set of effectors for host defense. The biological significance of the complement system has been recognized by the medical community, and now there are more than 10 complement inhibitors currently approved, with dozens more in clinical trials and development. In addition to therapeutic approvals, evaluation of complement system proteins is being included in clinical diagnostic guidelines across many pathologies. Research into the complement system has increased significantly in the last 25 years. Access to high-quality laboratory assays and reagents is key to understanding the role the complement system plays in numerous research areas. QuidelOrtho offers a wide variety of laboratory tools to assess complement proteins, including ELISA kits, ELISA-based multiplex assays, antibodies, proteins, and specialized reagents. These products have been validated by the complement research community across diverse applications and sample types in thousands of peer-reviewed literature publications. The complement system is vital for primary immune response and is associated with a variety of disease states. QuidelOrtho is your partner for complement research products to further advance this evolving field.